Alright, this is part 2 for your study guide short answer notes. I'm going to try to go faster than what I did the last time. That was a long video. So using the karyotype, explain whether this human individual is chromosomally normal or displays an aneuploidy. Is the individual male or female, and how do you know? Well, a normal individual is going to have a pair of each chromosome, and no more, no less. So if you look at this individual, you've got an individual with 2 of everything except for this chromosome number 21. So this is your aneuploid part of your chromosome, so it is not normal. Is it male or female? A male would have 1 X and 1 Y, but this is a female because it has 2 X chromosomes. So you would just state whichever karyotype picture you see and explain that. And I shouldn't say that in the example with the fur collar and rabbits you will have a similar dihybrid cross, but maybe not the exact same one to discuss in your answers. Number 7, explain how pedigrees are helpful tools in family planning. Of course, they allow you to see whether or not your individual has a chance of passing on a trait that is part of their family line. Whether it's a disease or a good one, you can kind of tell the chances of that. Using the pedigree below, is the indicated trait a dominant or recessive trait? How do you know? Well, if it's a dominant trait, chances are it's going to show up in every single generation. If it's a recessive trait, then you're going to have 2 parents that do not have the trait and an individual that does. That can't happen in a dominant trait for 2 parents to not have it and for an individual to have it. So that happens here, these don't, but this one does. And that happens here, these don't, but this one does. So in those 3 scenarios, you can tell that it's a recessive trait that occurs throughout the generations. Next question, is an X recessive sex-linked trait in one of these? Are there more males or females likely to exhibit the trait, and why? So in a sex-linked recessive trait, there are actually males are more likely to exhibit the trait than females, and that's because you inherit the chromosome from your from your mother, and you inherit the Y chromosome from your father, so there's nothing to cover up those chromosomal abnormalities or mutations or whatever you're talking about in males. In an XX individual, however, like a female, you have the chance of getting a dominant or a recessive trait on the other chromosome, and so it can mask whatever trait is there. This next question, based on the gel electrophoresis, which individuals are related? How can you tell? Use the term restriction enzymes, short tandem repeats, and electricity in your answer. So restriction enzymes are what cuts these DNA into fragments. They're looking at specifically 13 short tandem repeats in the United States to determine relatedness. And electricity is what moves these fragments through the gel. So larger fragments are stuck at the top, and shorter fragments move to the bottom more easily. So this is our example. You've got mom here. Mom has, Mom has these 2 right here and right here. That's what you're looking at for mom. So if an individual has that, it would be related to mom. This individual has one of these, it's related to mom. And so on. Let's see here. The father has this one and this one. Well, child C has this one from dad and has this one. Child D has that one from dad. So child G has information from dad and information from mom. Child C has information from dad and information from mom. And child E has information from mom and information from dad. So all of these individuals are related in a family. Next question. Explain how translocation during meiosis can result in a genetically normal or genetically abnormal individual. Well, translocation is where part of one chromosome can end up on another chromosome, so it really depends on how the non-disjunction or how the inheritance of chromosomes after metaphase 1 and metaphase 2 happens. If you still end up with all the parts of the chromosome that you need, even if they're on different places, you can still have a genetically normal individual. You would be abnormal, let's say, if you got only half of one chromosome but a full one of another. So for example, if I were to draw a picture, this is my lovely little cell, and let's say translocation, this is my normal chromosome 1, this is my, and its copy, And let's say that I have another chromosome here, but half of this chromosome got stuck over here for some reason when we split the first time. So this is going to look kind of funny, but after those split, you have this guy and its one and its extra copy, and then this guy with its half a copy, and this one, and then if these split again, you have a normal individual here, and this one has 3, so that would be abnormal, because this one should be on this guy down here, and then you have half of one and then a normal one. So in this situation, even in translocation, you can still have a 50 shot of having a normal individual. This one would have an extra chromosome, half, and this one would be missing half of its chromosome. So depending on which chromosome that was, it would determine the lethality or the success of the fetus. Next question. Describe the biology behind and provide an example for the statement genotype plus environment equals phenotype. Your genotype are the genes that make you up. The environment are what causes the expression or the hindrance of those genes. And then what you see is the phenotype. So the biology behind it is you can have an environment that causes methylation of your genes, and methylation would be the overexpression of those genes or causing them to be expressed a lot. So in the case of skin cancer, for example, you could have genes that are methylized. Well, I don't even know if that's a word, but you get the idea, that cause the cell cycle to continue and to not shut off. And in that case, your cells would continue to divide and not know to stop, and so those could cause cancer. Another case of this is just seasonal changes in fur color in animals. So depending on what the environment is doing, your animals would change their fur color or not. So a rabbit that would normally change from a brown fur to a white fur in the winter in a higher, colder environment, if the seasons aren't changing, they might stay a brown fur instead of change. Number 12, animals and plants both undergo cellular respiration. However, only plants can perform photosynthesis. How are the two processes related? These processes are related because plants and animals both do the cellular respiration. Of course, it says that, but the idea behind it is that photosynthesis reactants are the same thing as cell respiration products, and so they're the reverse of each other. So one gives the sugar that the other one needs to break down to make energy. Last question. In lab, we studied yeast. Describe the process it underwent to produce the carbon dioxide bubbles. So we made yeast make carbon dioxide because it went through anaerobic respiration called fermentation, and it made carbon dioxide and ethyl alcohol as a result of breaking down sugar without the presence of oxygen. And so normally when that happens, it makes pyruvate molecules, which are a 3-carbon molecule. But in the case of fermentation, it will make, what happens is it makes ethyl alcohol, and the other carbon that comes off of that is ethyl alcohol. It's a 2-carbon molecule, and it comes off in the form of carbon dioxide. So I'll relook at that.